Hello everyone, welcome to my channel. Today we'll be talking about Brightfield microscopy. So let's brighten up the way. We will be talking about the working principle, okay? The samples which will be useful for Brightfield microscopy, various advantages of Brightfield microscopy, and also the disadvantages associated with this technique. Okay, so let's start. First of all, I think all of you are very well versed with a microscopic configuration. So this is the light piece, and from the eyepiece, if you move down, then you have the objectives, and the objectives are positioned in a nose piece. Below you have the stage, then the condenser, and then the light source. Okay, so in this bright field microscopy, the light source can be anything. It can be a mercury arc lamp, it can be your metal halide light source, or it can be the very new and the latest one as the LEDs. Okay, so which is currently very popular. Now if you talk of LEDs, the heat associated with the LED will be very less, and the lifetime is quite more, so people prefer LED. LED also is a white light source. Okay, so this is something you should remember. So as this is white light source, you don't have to add any filter. ठीक है, तो आपको कोई filter लगाने की जरूरत नहीं है. You just have to use the light source. LED light sources even are very small in size, compact in design, so that is why it is growing interest in the customers these days. Most of the places you'll find the old traditional light source have been replaced with the LEDs. Okay, now this light source, or from the LED or whatever source, the light is coming, and you see the light is being focused onto the sample, right, after passing through the condenser. So the condenser's role is to focus the light onto the sample. आपका अगर पूरा लाइट सैंपल पे नहीं गिरता है तो क्या होगा, आपका लाइट आ तो रहा है but illumination is not proper. Okay, so illumination, if it is not proper, then what will happen is you will not find even illumination in the field of view. Some portion will be brighter, some portion will be darker, and that obviously is not your objective, right? So after the light is passing through the sample, now one small important thing, light has to pass through your sample. So the light source is giving light, that is true, but your sample has to be transparent. Okay, so if the sample is not transparent, light will not pass through the sample, and it will not be collected either through the eyepiece or by the camera you are using it. Okay, now if you see, this is the schematic diagram. So light, you see this light is coming out through the condenser, it is getting focused onto your sample, and once it is focused onto your sample, you have a focused illumination. The light passing through the sample goes to the objective, and from the objective through the ocular lens you can visualize this. So this is as simple. You can see the light path for a bright field microscope. Now what are the criteria you have to think of? See, light is passing. So first is your sample has to be transparent to light. Light has to pass through the sample. Okay. And if light is passing through, then only you can see it. Now, what is the contrast, or how you are generating the contrast? So what important thing is, if you see, the bright field is derived from the fact that the specimen is dark but the background is bright. Okay, and that is why this bright field name has come, nomenclature has come. This is pretty straightforward. There's nothing no important or nothing very significant or deep meaning into it. But if you say the sample is dark and the bright is the background, then will it have problem? Not at all. It will not have problem. The sample is dark because light is passing through the sample and some amount of light will be absorbed there. Okay, so there you see your sample little darker, but even if it is darker, you see it's a bright field, so there is a lot of light, there is a lot of brightness into this. Now when you have a lot of light and brightness passing through your sample, obviously the contrast can be slightly generated. When the light passes through the sample, there will be a slight light loss. Okay, applications, if you see, there are lots of application areas which you can think of. you can use simple blood smears. So if you have like a pathology lab, so you give your blood, you give your urine, you give your stool samples, right? So a pathologist will put it on a glass slide and they stain it with common stains, Brightfield stains, okay? And you see different colors attaching to different body. So if you have a first one, if you see, this is from the blood smear, so you see different types of cells like RBCs, WBCs, and what are the different types of WBCs are present, you can find it. So for pathologists, for screening purposes, this is very commonly used, okay? It's like a lab next door to your house. You go there and you find a small microscope, they're working in Brightfield mode, okay? In addition, you also have like plant samples. So you have root, you have leaf samples, you cut them into finer sections, you stain them, and then you see it. You can see a lot of colorful, xylem, phloem, vesicles, cell wall, cell structures, everything is quite easy to see. For histopathology samples, okay, now if you're coming to, again, this is a part of pathology again. So histopathology samples, they have tissue sections. So in the tissue sections, they simply have to stain it and put it on a glass slide. Once they put it on a glass slide, what they see is, you can see here, this one, you see the tissue morphology. So you say the cells which are normal cells, cells which are not normal in the types of morphology, in the types of distribution, in types of cell-cell interaction. So interaction means it is a fixed one, it's dead, it will not have any movement, that's true. But at the same time, if two cells are there and one cell is not healthy, you can see from the morphology. So the neighboring cells will also be different from this particular. So you can count, like if, for example, a ERPR, if you have to count that. If you have CD4, CD8 marker cells, the marker cells will be stained darker, the healthy cells will be stained lighter. You can count the percentage of your marker cells compared to the healthy cells. So all this are histopathology part, and it is, I think, a very routinely done in a histopathological lab. And you go to any pathology where histopathology labs are there, and you will find this being done at a regular routine work. Okay, now there can be also like algae, sea anemone on small like voucheria. So this small water plants or algae is basically, you can put it, like you can see here, one small one, you can see. Now interesting fact is they have a lot of chlorophyll. You see, they have a lot of their inherent color. So these colors are visible to the eye. That is why you don't need to stain most of them, and you can easily put them on your sample, let the light pass through it, and it is as visible to your eye. Similarly, you can record it with the help of a camera. Okay, now the small example, this is a green algae, and this is called cladophora. So these cells, if you see here, in this you see you can see small, so these are cells. So if you see this cell compartment and how one cell is divided from the other, what is the basically the morphology, and how this and you can see the there's a lot of chlorophyll inside, you see a lot of green color inside. So this is not being stained, but you can simply use this Brightfield just to see that inherent color of the sample. That is also a one big USP. Don't have to stain also always, you can use it. Now after staining, this is one example of a gram staining. So this is gram stained bacteria, and you can see the culture samples. So you have again, coming to the pathological applications, where your sample has come, they culture it and they do staining. Gram positive, negative stainings are being done, and then based on that they count it whether the infection is there or not. Okay, so next time when you give your sample to any pathological lab, think, is the microscope being used in the process? And I would say yes, yeah, right. Okay, so interesting part, one more, or how you're getting the contrast where there is no contrast is by staining, right? So when you do staining, there are different types of staining techniques available, or you can say Brightfield stains available. So for example, you have crystal violet, okay, you have like methylene blue. Okay, so there are a lot of this small acridine orange. So this all different stains are which is very commonly available, and what you can do is you use these stains, put it in your sample, and once you put it, you get a lot of this colors goes and fix onto the architecture of the required. So they will not go and bind everywhere, but they go on specific target, and then wash it off. After you wash it off, your backgrounds become clear. The stains have already touched on those areas where it is meant to be. For simple, I'll tell you. Suppose I am wearing a shirt, white shirt, and I am wearing also a cap. Okay. Now, this is holy time, and you know people use a lot of color balloons. So somebody has dropped color onto me, but I have one specific thing. This particular color can stain either my shirt or my cap. So if I have two different colors and one color can stain either my cap or t-shirt, so at least when I'm colored with two different colors, I'll have cap of one color and t-shirt of another color, right? So even if I'm using, so my t-shirt is suppose blue and my cap is red, even if you're using red, my t-shirt will never be red, and then I go and take a wash, but my t-shirt color remains blue, my cap color remains red. So this is specific. So when a person, I tell another person, okay, you know, if you see red, it is a cap. If you see blue, it is a t-shirt. So similarly, when you for your sample also, the stains will go and bind to specific parts, and then you wash it off. The background becomes clear, and your specific part of the sample will be stained with a particular dye, and that is how you separate it out. Okay, interesting, right? Yes, it is. So when you practically do it, I think it will be much more fun. You can get in your in your home also. You can have lot of like leaves. You can collect leaf, add some, you know, even your blue color dye, so ethylene blue and all. This is commonly available. Acridine orange, they're very commonly available. Just add a drop of it, then pour some water, rinse it, oh, let it wash, and then you see it against any, even a magnifying lens also, you'll see the different colors and it will look beautiful. So you know you can do that. Yes, but advantage, yes, of course, you have lot of, like, it's very simple. As I know, it sounds fun when I'm talking to you, isn't it? So you just take the sample, drop one, give a washing, and you can do it. So one simple example I think in school we have done is cheek swab. So you take a swab, and then you put it in your mouth and take it from the cheek side, roll it, put it in the glass slide, add some dye, and then you can see the cheek cells under any simple microscope, and that is a Brightfield experiment. Okay, you don't need much adjustment, light is enough, so it is very simple. But yes, limitations are there. uh, you know, as it is going and binding, specificity is little low compared to a molecular specificity or target oriented specificity. Here specificity is pretty low. It goes and binds to overall areas in a larger areas. Contrast, at times it is difficult. It is like, suppose you are working with diatoms, you will not be able to do with Brightfield. So in that case, the staining is not possible. Suppose you are working with flagella and or for example bacteria, even if you have staining it, getting a resolution, and when working with Brightfield will be a little difficult. You will not get all the details out of it. Okay, and obviously you're using a strong light source, so at times your sample may not like it. Even if you're working with the live cell lines, your sample may not like a brighter light. Okay, so in those scenarios, it's a no for Brightfield. But of course we have other techniques which we are going to discuss in later sessions. So a few references, here are the few references, please go through them, read. I would request all of you, please like, share, subscribe. If you have any comments, feel free to get back to me. You can comment, you can, you know, even mail me or even WhatsApp me. Thank you for your support.